Bismillahirrahmanirrahim. Assalamu alaikum students. Today we will discuss stem anatomy in Ephedra. Stem of Ephedra, it is wavy in outline in transverse section because in the stem ridges and furrows are present. The stem shows the following parts. Number 1 is the epidermis. Epidermis is the outermost layer which consists of single layer of cell, and these cells are parenchymatous and rectangular in shape. And the outer walls of these cells, these are covered with cuticle, and stomata in the epidermis are present in the furrow regions, and these are sunken stomata. Second part of the stem is hypodermis, which is present below epidermis. This hypodermis is not continuous, and only beneath the ridges, means below the ridges, patches of elongated sclerenchymatous cells are present, which have thick lignified walls, and these form the hypodermis. And this hypodermis region, it provides the mechanical strength to the stem. Here in this diagram we can see this is outermost epidermis layer, and below the epidermis, below the ridges, sclerenchymatous part is present. Patches of sclerenchyma cells are present below the ridges, and in the grooves or in the furrows, stomata are present, and the wall of the stem, it is wavy due to the presence of the furrows and the ridges. ये ridges है जो उभरे हुए ऊपर की तरफ parts है, and these grooves are the furrows. So alternate ridges and furrows are present. Below the ridges, hypodermis is present, and below in the furrows, stomata are present. We can see here the part of the stem. Here, the outermost layer, epidermis, the cells of which are parenchymatous and rectangular in shape. The outer walls of these epidermal cells, these have thick cuticle. Thick cuticle is present in the outer walls of the epidermis. Then below the epidermis, hypodermis is present, and the patches of hypodermis are present below the ridge region. This is the ridges, these are the ridges, and in this region below the ridge, the patch of lignified sclerenchyma cells are present, and in the furrow regions these are stomata which are present, and these stomata are sunken. Then the third part of the stem is cortex, which is present below the hypodermis region. And cortex in the ephedra, it is differentiated into two zones. The outer region is composed of elongated, loosely arranged palisade-like cells, and these cells contain stomata. These cells contain chloroplasts, and these cells are photosynthetic due to presence of chloroplasts. The inner zone is formed of oval, thin-walled cells which resemble in shape to the mesophyll cells of the leaves, and these cells also have chloroplasts. So the internal structure of the cortex, it resembles to the structure of the leaf. Below the hypodermis and epidermis, this is the cortex region, and this cortex is differentiated into two regions. The outer zone consists of elongated cells which are palisade-like, and these contain chloroplasts. And the inner zone, it consists of also the parenchymatous oval-shaped cells, and this region also contains chloroplasts. Then in this region, in the cortical region, intercellular spaces are also present, and this cortical region of the stem in Ephedra, it resembles to the leaf mesophyll tissue. Then below the cortex, the innermost layer of the cortex, it is differentiated into well-developed endodermis in young stems. Here we can see the innermost layer of the cortex which forms the endodermis, and then next to endodermis vascular tissues are present, because in Ephedra below the endodermis pericycle region it is not distinct. So next to endodermis the vascular tissues are present, and vascular tissues in the Ephedra, these are present in the form of vascular bundles, and each vascular bundle, it is conjoint, collateral, endarched and open, and vascular cambium is single layered, and this we can see here below the cortex region. These are the vascular bundles, and these vascular bundles, vascular tissues are in the form of vascular bundles. Distinct vascular bundles are present. And in these vascular bundles cambium is also present, and these vascular bundles, these are present in the form of rings, and between the two vascular bundles the spaces which are occupied by the parenchyma cell, these form the medullary rays. These are the, this is vascular tissue. Vascular tissues are in the form of vascular bundle. This is vascular bundle. This is, these are also, these are vascular bundles which are arranged in the form of ring. These vascular bundles are conjoint, mean that phloem and xylem are present in the same radius, on the same radius, in the same vascular bundle. Vascular bundles are open because cambium is present in between the phloem and the xylem. This is the cambium which is single layered and present in between the phloem and the xylem, and the space which is present in between the two vascular bundles, this is occupied by the parenchyma cells which form the medullary rays. The xylem and phloem which are present in these vascular bundles, these have different components. The xylem vascular bundle, these consist of three components: tracheids, number 2 vessels, and number 3 xylem parenchyma. The vessels which are present in the ephedra, these originate from the petite tracheids which are present in the ephedra. Xylem and the tracheid walls, the walls of the tracheids, these show thickenings which are annular and spiral, and on their radial walls bordered pits are present. And in the vessel members, the perforation plates which are present, these are foraminate type, and on their lateral walls also bordered pits are present. The presence of the vessels in the gymnosperm is the character which is present in the angiosperms. means in angiosperm xylem vessels are present, and these vessels are also present in the ephedra of the gymnosperms. Like xylem, phloem also consists of the components number 1 sieve cells and phloem parenchyma and albuminous cells, and center of the stem it is occupied by the parenchymatous cells which form the pith region. In this diagram we can see here again the vascular bundles in ring, medullary rays which are present in between the two vascular bundles. This is center of the vascular tissues, this which is occupied by the parenchyma cells which form the pith region. Then in the stem of ephedra, secondary growth also occurs due to the activity of the cambium which is present in between the xylem and phloem of vascular bundles. Before the secondary growth, the cells of the medullary rays which are present between the vascular bundles, these become meristematic and they join the vascular cambium of the vascular bundles to form a cambium ring. In this way, a circular continuous cambium ring is formed. The secondary vascular tissues, these are produced in the form of distinct annual rings, as the xylem which is produced in the early season and the late season, both these continue to develop or to form one annual ring, and in this secondary growth more secondary xylem is produced than the secondary phloem, and during this secondary growth secondary medullary rays are also produced. The most important feature of the secondary xylem is the formation of the vessels by proliferation of terminal walls of some of the tracheids, means the vessels which are present in the secondary xylem of the ephedra, these are formed by the expansion of the terminal walls of the tracheids, means some of the tracheids, their terminal walls they are expanded, and as a result of this the vessels are produced. In this diagram we can see here the production of the cambium ring during secondary growth. The cambium ring which is produced by the production of the cambium in between the vascular bundles, and in this way a continuous cambium ring is produced, and this cambium ring on the outer side it produces the secondary phloem, and on inner side it produces the secondary xylem, and primary xylem is pushed towards the center in the pith region. And in the secondary xylem some of the tracheids they are converted into the vessels, and the presence of the vessels it is the characteristic feature of the ephedra stem, and in the center pith region is present. As a result of the secondary growth in the vascular bundles, in the vascular tissues, pressure is exerted, and as a result of this periderm is also produced in the stem region. Here is another diagram showing the secondary growth and the production of the vessels in the xylem, secondary xylem tissue, and which is the broad region in the xylem of the ephedra stem. This is the primary phloem, the cells of which have been converted into the fibers. This is secondary phloem, cambium ring, and on the lower side of this cambium ring, secondary xylem is produced, which is more than the secondary phloem, and some of the tracheids which are present in the secondary xylem, these are converted into the vessels, and primary xylem is pushed towards the center. And in the center of the stem, pith region is present. You can see here again the diagrams of the young stem. Epidermis region, hypodermis below the ridges, then cortex region, endodermis, and then vascular bundles. In the vascular bundle, this is phloem, this is xylem, this is cambium ring, and this, as a result of secondary growth, this cambium ring, when it becomes continuous, it produces the phloem on the outer side and xylem on the inner side, and in the broad xylem region, vessels are produced, and on the upper side of this the stem, periderm is also produced, which is also a tissue, protective tissue, which is secondary in nature. And this is the diagram of young stem in which the epidermis, hypodermis, chlorochromatous region, cortex, and vascular bundles are shown along with the pith. And this is the part of the xylem tissue, which is showing the large number of vessels in this secondary xylem. So students, today we have discussed the internal structure of the stem, in which we discussed that stem of the ephedra, it consists of the parts: number 1 epidermis, number 2 hypodermis, number 3 cortex, number 4 endodermis, and then vascular tissues. And the main thing in the anatomy of the ephedra stem is that vessel members are present in the secondary xylem of the stem of ephedra, and this character it resembles the anatomical character in the angiospermic plant.